Welcome to this, the sixth of our 9-part series of the Origin Science Scholars, presented by the Institute for the Science of Origins, a partnership of Case Western Reserve University, the Cleveland Museum of Natural History, and IdeaStream, with the assistance of the College of Arts and Sciences and the generous support of Richard Morrison. This week's speaker is Dr. Darin Croft, Associate Professor of Anatomy at Case Western Reserve University's School of Medicine, and he will talk to us this week about species, speciation, and evolution on islands. Dr. Croft.
Thank you very much. Before I get started, I do want to put on a little bit of a plug. Speaking of the museum, Natural History Museum, there's going to be a great exhibit that's going to be opening up in a couple weeks. And since I won't see you again for a couple weeks, I figured I'd mention it on extreme mammals. And I'm going to show a few extreme mammals at the end of this talk and probably somewhere in between. So if you like cool mammals with big teeth and lots of headgear, I'd recommend you check that out. I think it opens beginning in November and it'll be there through the spring. So I wanted to say that now because at the end I always forget about things like that. The game plan for tonight is really to follow the title. We're going to start talking about what is a species, talk a little bit about how they form, and then finish up with evolution on islands, because it turns out that islands are really great places to isolate some of the different processes that are affecting speciation. And they're one of the things that really got me interested in evolutionary biology and paleontology, and we're going to finish up with that, which I think will be fun. Hopefully you'll agree. First, let's talk about what a species is, and we have to do a little bit in terms of definitions. There are two different ways that we can talk about a species. I think it's a term that everyone uses all the time. Everyone uses species, knows what a species is, but there are really two senses that we use it, and we don't usually differentiate. One is in a taxonomic sense, and this hierarchy that I have there you might have learned back in biology: kingdom, phylum, class, order, family, genus, species. The main levels by which we classify different organisms. It's like a Dewey Decimal System for animals. Right? We have a big animal kingdom, and then we could talk about different phyla, like things with backbones and jointed animals. We could talk about classes and orders and work our way down from there. And at the very bottom of this level, we have the species. It is therefore a taxonomic name, and everything has to fit in there, because this is our organizational scheme. Just as we can't have a book that doesn't fit in the Dewey Decimal System, we can't have an animal that doesn't fit in this scheme. So every animal is a species, even though we have little things like this, which reproduce asexually, like a bacterium, which biologically speaking may or may not be a species depending on our definition. And we're going to talk a little bit about definitions. So when I'm talking about species, for the most part, at least for the next first part of this talk, we're going to be talking about this biological sense: what is a species, not the taxonomic term. Keep in mind one or both may apply. Definitions. There are a variety of ones we can choose from. We're going to start with this one, the biological definition: groups of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. This was coined by Ernst Mayr back about 70 years now, and this is one of the oldest definitions and certainly one of the most popular. And I want to call your attention to a few different parts of that. One is, it talks about actually or potentially interbreeding populations. We'll hit on that a little bit further down the road. And specifically, we're talking about natural populations. This is not a definition that we're going to apply to things like domestic animals necessarily, talking about whether or not they're species. And finally, reproductively isolated. And that's really the crux of this biological definition. And we talked about this a little bit a couple weeks ago in the question and answer session: what is a species? And I think hopefully you got a sense that there's some dispute, depends on your definition, so this isn't all new to you. But the real crux of this one is reproductive isolation. And this is the most widely used definition, partly based on the perspective of the person or the people who are using it. And I'll hit on that again too. The key thing that I'd like you to remember is that it is process-oriented. Thinking again back a couple weeks, we talked about evolution, and my argument was that evolution is a pattern, and we have a process that accounts for much of that pattern, that being natural selection. But we can also have random genetic drift. We can have also all sorts of other things. It's sort of analogous with this. This is a process-oriented definition, and it's really focusing on the process of reproduction, which, as you can imagine, can be difficult to get at sometimes. How about some alternatives? There's a phylogenetic definition that's out there. Species is an irreducible cluster of organisms that is diagnosably distinct from other such clusters, and within which there is a parental pattern of ancestry and descent. This is the way that Croft characterized it back in 1989. We're not going to go through this with a fine-tooth comb, but the crux of this one really is diagnosably distinct. Something's diagnostic. When we name a species taxonomically, we have to give it a diagnosis, which is the way that it can be identified relative to other species. And that's really what this hinges on, is this diagnostic ability. Therefore, it is a pattern-oriented definition, quite different from the biological definition. Mostly they're going to overlap, but there's some places where they won't, and where they'll come up with different decisions about whether or not something is a species or how many species there might be. A recent one that was just summarized a couple years ago is a genetic one. It's similar to the phylogenetic that we just saw. Defines a species as a group of genetically compatible interbreeding natural populations that is genetically isolated from other such groups. And that's the key with this one, genetic isolation. It also is pattern-oriented, but it's looking at a different part of the evolution equation. Evolution occurs on a genetic level, right? Thinking again back a couple weeks, we talked about inheritance. It has to be inherited variation. And I know Cynthia talked a lot about this too. If you're not inheriting it, you're not going to get evolution. But we think for the most part, natural selection is acting on the phenotype, which is what something looks like or other characteristics. And so we have two definitions that are looking at different aspects of this. And this one's focusing on the genetic difference. There's evolutionary species concept, there's a genealogical species concept, there's a recognition species concept, there's a cohesion species concept. We could go on and on. There are lots and lots of different species concepts out there. And you could probably come up with one of your own. The key thing in my eye is that a definition depends on an author's reasons for defining a species. A definition is a tool. And depending on what your research is focused on, you're going to find one sort of tool more suitable than another sort of tool. And that's why all these different ones exist. And of course, some are more popular than others, but they all have their issues, which we can call limitations. And here's one example, domain of application, which is a fancy way of saying to what organisms does it apply, or what sorts of organisms can you classify or define with that particular definition. And of course, we already showed one example of this. Single-celled organisms get to be a little tough when you talk about reproductive isolation. But I'd like to call out another one, being a paleontologist. I know there are other paleontologists in this room. This is something that we struggle with. We've got a time axis here, just like we always put it in geology, with old at the bottom, young at the top. And we can look at extinct organisms and a phenomenon known as anagenesis. And anagenesis can be drawn something like this. We have a population that has some sort of morphology. We move through time. We have another population that is shifted from that original morphology. We move through time again towards the top. We have population C that looks a little bit different. If we think that this is one population that is just changing through time, then that's what's known as anagenesis. Just basically change in a lineage. What gets challenging is deciding whether these different populations are different species. And that depends on our definition. If we are applying a biological definition, it is a challenge, because we haven't yet figured out how to determine whether fossil species are breeding with each other. And this is a dimension that we as paleontologists deal with a lot, but biologists don't. And so we as paleontologists often have to modify the definition a little bit into something that's more pattern-oriented. And that's why you often see that something like the phylogenetic definition is going to be something more used by a paleontologist. And what it really comes down to is morphology, because that's what we have in the fossil record for the most part. Get recent fossils, we can get into DNA, but mostly we're dealing with bones. Thinking back to our old friend Ectosion, that early ungulate lived in the late Paleocene, early Eocene, about 55 million years ago, right when we had that big temperature spike, that Paleocene-Eocene thermal maximum. This is the same slide, essentially. And this is a real-life scenario that we have to deal with. What do we do with that species down there in the bottom? We've got E. parvus, it's that small one, right at the thermal maximum, where its teeth got really small. Presumably the rest of it did too. And E. osbornianus up above. But what do we call that one on the bottom? And it's interesting to think about what would happen if we didn't have real fine resolution and we didn't pick up that little blip of E. parvus. We might call that whole thing E. osbornianus. But we don't really have a model that accounts for a species becoming another species and then going back to the same species. So we almost have to call that something else. Or maybe we can make an argument there's a little point there that's right to the far right here. Maybe that's E. osbornianus hanging out. Maybe there's just not many, and it's actually outside the range of variation. These are all issues that we have to deal with. Problems with the morphological definition. So no matter what definition you use, there are challenges that you have to overcome. Another limit is borderline cases. Here's a nice one with plants. Not sure how green those look. Those look nice and green on my screen. These are different oaks that live down in the southwest U.S. Gamble's oak on the right side and the gray oak on the left side, which are different species. But we get hybrids in between. And you can see that morphologically, they are intermediate. Right? We have a small-leafed gray oak and a very large-leafed Gamble's oak that has lobes. And we get variations in between there. Depending on whether you're using a morphological definition or a biological definition or potentially a genetic definition, you might come up with different schemes for this. And you might not call those hybrids. You might say that that's actually a distinct species. Depends if you're the botanist studying those, which you might prefer. Practicality. This is a good one. There are lots of things we'd like to do as scientists, but it's just not practical to do it. Mountain lion, Felis concolor, is one of the most widely distributed mammals, certainly in the Western Hemisphere. It ranges from the far southern part of Alaska, almost, all the way down essentially to the tip of Tierra del Fuego. And there have been genetic studies that have been done on this and morphological studies. And the bottom line now seems to be that we don't have 32 subspecies, like we once had named. That really is just one population. But I would be willing to bet that the Canadian pumas are not breeding very often with the ones down in Buenos Aires. Except maybe some of the politicians, I don't know. Perhaps they fly down there for that. But regardless, if we're using a biological definition, we have to ask, are they reproductively isolated? And the practicality of testing this is such that we're not really going to test that, and we assume that other proxies can stand in for that reproductive isolation. We assume that if they're genetically distinct, if they're morphologically distinct, then they are not interbreeding, but if they're the same, then they probably are. It's just not practical to test this explicitly. The bottom line for what is a species. First, there are many different species definitions. Other ones I could have put up here that I didn't put up there. I think they're enough to illustrate the point. No definition is perfect. So don't let anyone tell you that one definition really is the answer and that it fits for everything, because we haven't found one yet that does fit for everything. Most work for most things, but there are certain cases where they're going to lead you to different conclusions. And the optimal definition really depends on the circumstances. If you're dealing with paleontology, you'll probably be using a different definition than if you're working with an ecologist who might have a different definition than even a botanist. And that's all we're going to say about species. Moving on to something that's a little more process-oriented, we're going to talk about how species actually form. How do we get the biodiversity that we see around us today? I have a few books that I'm going to put plugs in for in this talk. One is this one, which is on speciation. There's enough to write a book on, suffice it to say. And this is a pretty recent one. It's about 2004. So if you're interested in learning more about the process of speciation, species definitions, any of the issues like that, I'd encourage you to check this one out by Jerry Coyne and Alan Orr. Okay, When we talk about how species form, there are really two main ways that we can classify the process. One is a biological way, and the other is a spatial way. Ultimately, speciation is the result of biological divergence. Evolution is a biological process, so speciation also is a biological process. And we talk about these different isolating mechanisms that cause divergence between populations. However, such divergence can occur under different geographic conditions. And this then is another way that we can classify speciation. These are not mutually exclusive. They're just different ways of looking at the same process. We can therefore speak of both biological isolating mechanisms and of the spatial contexts in which the species form. And I'm going to give a little bit of an overview about biological isolating mechanisms. There's a deep, deep literature about getting into chromosomes and genetics and all those sorts of really baseline, fundamental isolating mechanisms. We're going to talk a little bit more on a gross level in terms of those, and we're going to start off by discussing those. The biological isolating mechanisms we can also sort into a few different parts. Basically, those that keep people from or keep animals, and probably people, I guess, from mating. Those that cause problems during the mating, and then those that cause problems once fertilization occurs. A nice example of what we could call a pre-mating isolation is something that we see in great cats, like tigers on the left there and lions on the right. And these are ecological barriers that prevent mating. So we said pre-mating isolating mechanisms keep the mates from coming together, basically ships passing in the night. Tigers are mostly closed habitat carnivores. They like dense jungles. That's where they hang out. Lions, open habitat. Right? You think African safari, you think lions. For the most part, on a broad geographic scale, these aren't overlapping. But their ranges do overlap in a small area in India, and they intersect. In theory, you could get tigers running into lions, perhaps getting along very well, perhaps mating, and producing a liger, or I think a tiglon is the other one. And we know this happens because they can be produced in artificial circumstances, right, at circuses and zoos and things like that. But it doesn't seem to happen very often in nature, and the reason we think it doesn't happen is because of this ecological differentiation. The tigers, even where their ranges overlap, are mostly in the denser habitats, and the lions are mostly in the open habitats. So although on a gross level they're overlapping, on a smaller level they're really not hanging out in the same place. So this is an ecological, because it's attributable to habitat, pre-mating barrier. Keeps the mates, potential mates, from coming together. It's like hanging out in different bars, essentially, right? This can also be behavioral. I don't only look at mammals. We're going to include a few other things in here. And this happens to be a lacewing. If there are gardeners among you out there, lacewings are good insects. They are helpful. They're predators. They eat things that eat your vegetables. So if you ever see lacewings, you don't want to kill them. It's a good reason for trying to restrict your pesticide use because they take out the lacewings as well, of course, as the things that are eating your vegetables. But so much for that. They call, just like crickets, just like a variety of insects, katydids, to attract mates. And different species can have different calls, and that's what we're looking at right here. These are sonograms on the right, and you can see that their calls are of different frequency. C. johnsoni is only making 3 calls in the same period of time when C. pluribunda is making, I don't even know how many, a dozen or more calls. Because there's a positive relationship to these calls from females of the same species, this tends to prevent cross-species matings. So a female of one species won't be attracted to the call of a male of another species. They're both living in the same areas, even on a fine scale, but this is a behavioral issue that's keeping them from coming together. It's still a pre-mating isolating mechanism, but it's behavioral rather than ecological. How about with the actual act itself? We get some animals that come together which may or may not be of the same species. we can get post-mating, pre-zygotic isolating mechanisms. And it's really kind of a mouthful, but basically it means we get mating, but there's something that prevents fertilization from taking place. We'll classify these as copulatory issues in this example, and these are rodents. Rodents are very successful as far as mammals go. They're the most diverse group of mammals. About 40% of all species are rodents. They're diverse, their morphology is diverse, and their bacilli are diverse. If you don't know what a baculum is, you're looking at a bunch of them on the left. They are penis bones that obviously are only found in the males of the species. This is something that humans lack, but they're found here and there throughout the mammal family tree, carnivores and in rodents, and they can be useful for diagnosing different species. On the right, we have the soft tissue. Those ones on the left happen to be from squirrels. The ones on the right are from a diverse group of essentially mouse relatives. There's a special group that are only found, at least for the most part, in the Western Hemisphere, and a lot of them have diversified in South America. And each of these are very close-up shots of a penis of one of these different species. And that little trident-shaped thing in the middle is the baculum. Mechanisms like this can act to prevent successful mating, or at least successful fertilization, even if two different species come together. We see this in bats, we see this in rodents, we see this in all sorts of different groups. So this is an example of how if species are different, even if they're in the same area, and even if they get to the point of mating, you may not get fertilization. There are lots of other examples too. How about we assume that two species come together, they're in the same spot, they mate, we get fertilization. What happens then? There are also a variety of post-zygotic isolating mechanisms. Something that either prevents the embryo from developing, or maybe it develops all the way to an adult and it just doesn't pan out in terms of creating a new species or even contributing to the gene pool of one or the other. And one classic example of this are mules. Right? You take a donkey and a horse, and you breed them, and you can come up with a mule. And mules have some of the good characteristics of both. They have some of the bad characteristics of both, but they're very popular as far as pack animals go. They are sterile, however, so you can't start breeding a line of mules. This is an example of a post-zygotic mechanism, and it's not uncommon. We could call this hybrid sterility. You get two animals that come together, they hybridize, and it's sterile. And this is what keeps these two as separate species. Although, of course, these are both domestic ones that we're using as an example, but you get the general idea. In the case of lions and tigers that we showed earlier, they can have fertile hybrids. So they don't have this problem. So there are probably other mechanisms at work to maintain those two species, in addition to the ecological ones that we talked about before. The previous example was an intrinsic issue with a hybrid. In other words, there's something wrong with the individual. The hybrid is fertile, or it doesn't work in a biological sense. We can also talk about extrinsic factors. Perfectly good animal, but something about the environment keeps it from successfully contributing to the gene pool. And this example really is an opposite of what I should be showing, but I think it's kind of fun, and that's why I included it. The Galapagos finches. And I know you've heard a lot about the Galapagos finches from Cynthia. It's an extremely well-studied example of diversification, of adaptation. We've got genetics on them. All sorts of interesting studies that have been done, primarily by the Grants and their collaborators. But one thing that's kind of cool about the Galapagos finches that I was just reading about, was a review article that came out this year. Turns out that under certain circumstances, the hybrids among these different species, right, we're mostly looking at beaks here. Beaks are where it's at in terms of bird adaptations. Under some circumstances, the hybrids have a higher fitness than either the parent species do. And sometimes they're not only surviving, they're surviving better, and they're contributing back into the gene pool. So we can envision a circumstance where that's not the case. And this is maybe what we think would be the case on the Galapagos. We've got a big beaked form and a small beaked form, and they breed together. Let's assume we get blending in a medium beaked form, and it's not very fit. So that's what keeps those as two different species. Here we have the opposite, and we don't have that impediment. And it turns out the hybrids, at least on occasion, are contributing back to that gene pool. And so the fitness of the hybrid has to do with extrinsic factors of the environment. It's not always, it's at some of the time. And that really gets back to a point that I'm sure that Cynthia hammered home, that being that fitness depends on your environment. And there's really no such thing as an absolute fitness, right? Unless you're in an environment that doesn't change, which is not true, at least on Earth. And it's an interesting example of where we could have an isolating mechanism, but we don't. Let's shift a little bit now and talk about the spatial contexts in which speciation can occur. And you might be familiar with this. Is something that's taught oftentimes in biology, and there's some terms we'll use that might be familiar. We can talk about speciation where the species are forming in different places, where they're forming nearby, or where they're forming in the same place. Where they form in different places is known as allopatric speciation. The patric in that part comes from patriotism, fatherland, and allo meaning other. Right? If you have an allograft, you're getting tissue from another person. So allopatric speciation is where the two populations are physically separated and speciation is taking place in two different spots. We can talk about vicariant allopatric speciation. And this is really the classic example. So we have one yellow blob up there, which represents a single interbreeding population, and then we get the formation of some barrier. Let's say it's a river, or maybe it's an ocean, or who knows what. Maybe it's a highway that's constructed between two populations. Those two populations, once separate, are free to evolve along different trajectories via natural selection, via genetic drift, via sexual selection, via all these different mechanisms for evolution that we've talked about before. If they come back together later in time, we will have two overlapping species, but it required this barrier for them to evolve. We think this is a way that species form often. Another way that speciation could occur that may occur frequently, sometime it's a matter of scale or definition, is what's known as peripatric speciation. Peri being essentially next to. We have a large, broadly distributed population, let's say the puma, for instance, and we get an isolate, a little group that pops off to one side that isn't necessarily geographically isolated, but they're perhaps in somewhat slightly different ecological area. So different factors are acting in terms of natural selection. It can evolve on its own, and then it can rejoin or at least re-overlap that main population, and we have two species living in the same spot. These are really just matters of differences in terms of scale and how sharp that barrier is that prevents gene flow between the two. But that's the bottom line. The gene flow is prevented because they're physically in different places. And we think this accounts for the vast majority of speciation. We talked about pumas. Here's a nice example. You could talk all night about it. We'll talk a little bit more later about it. But the Falkland Island wolf, or the Islas Malvinas wolf, if you're Argentine. These are the islands that are off in the Atlantic, off the coast of Argentina. This is a population of essentially fox that got off to that island. It was separated from its original population and evolved on its own. It's in a different spot. It's therefore allopatric speciation. Often what we see on islands. Nearby places. We can envision speciation occurring very close by, and this is what we call peripatric speciation. We have a single population. For some reason, we get divergent selection in the middle of that, which causes the two parts of the population to go in different directions. They eventually are isolated enough such that when they come back together, they're not interbreeding and they're actually two species. This one's a little bit more difficult to envision, but we can come up with real-world examples for this. And a classic one is in this grass, Anthoxanthum. And it grows both in its natural conditions and on mine waste. Hopefully you can read it. That's what it says there on the right. So we have mines that are dumping their waste out. We get populations, or at least individuals, of this broadly distributed population that are selected that are able to survive on that mine waste. This creates a downscale cascade of events such that the ones on the mine waste are preferentially breeding together. separate from the other ones, and you eventually are getting speciation. I do want to put in a plug for the site where I got this picture in and a couple other ones, because it has a lot of nice vignettes about evolution. They've got an Evolution 101 section. It's at Berkeley. So it's just evolution.berkeley.edu. So I'd suggest check that out at some time next time you're in front of a computer and want to learn more about evolution. Lots of great examples there. Finally, we can talk about speciation in the same place, in the exact same place, which is even a little more difficult to imagine. And this is sympatric speciation. Sympatry is together, allopatry is apart, and then para is sort of somewhere in between. And this is still highly debated, whether or not sympatric speciation really takes place. I think there's some good examples that it probably does, but it's certainly not common. If you want to read more about that, definitely check out that speciation book. So we have one population, and we get divergent selection acting on different individuals within the same population. Enough such that eventually we get the formation of a separate species through those biological isolating mechanisms in the exact same spot. Maybe you can come up with examples, but it's a challenge. Here is a pretty good one, though. This is a apple maggot fly. Not a great name, but what are you going to do? It's a fly. They eat apples. There's the genus name for you if you want to know. These are native to North America, where they used to feed on a native plant known as a hawthorn, which is very similar to our domestic apple. When apples were brought over, some of these individuals preferentially fed on the apples. From there, we got one of these positive feedback loops, sort of like we had with the grass, where females would preferentially mate with males who also fed on that fruit, and the larvae then, of course, would grow up eating that fruit and would preferentially go back to that fruit. And because all these fruits are scattered in the same area, as are the flies, this is an example of how speciation could occur in a single population. But you've got to have just the right circumstances to do this. It's a lot easier to just separate out the populations. And that's more or less how we think species form. We're going to finish up by talking about evolution on islands. And the reason that I chose to loop this into the species and speciation issues is because islands are really great for isolating a lot of the mechanisms, a lot of the causal mechanisms for evolution. And they've been enlightening for a lot of people and inspirational. And so I'd encourage you to keep these things in mind that we've talked about already as we go through these different examples of evolution on islands. A great book in my estimation, and one that really got me interested more in biogeography, was this Song of the Dodo by David Quammen. And it came out in I think the hardback in 1996, the paperback in 1997. It's a great read, talks about different islands. He has an excellent style. Some of you may have heard him speak here at Case a few years ago. He has that very nice biography, essentially, on Charles Darwin. And a great writer. And it's chock full of information. It's not a quick read. It's a thick book. So check that one out if you like the last part of this talk. Okay, islands. I like to classify things. Sometimes it works, sometimes it doesn't. But we're going to classify islands too, but hopefully not too much. There are really two different types of islands: oceanic and continental. Oceanic islands form de novo in the middle of an ocean with no species, and the only species that get there that are terrestrial are coming from somewhere else. Continental islands include a variety of different types, some that are separated through plate tectonics, some that are sitting on a shelf and are periodically isolated and then periodically joined back up, and it's a much more diverse group. We've already talked a little bit about oceanic islands with Hawaii. But there are lots of other ones out there too. The Galapagos, the Seychelles, at least that's how I pronounce it, it's probably wrong. Rapa Nui, otherwise known as Easter Island. Most of these are in the Pacific, partly because the Pacific is a big ocean, and there's lots of volcanic activity there. A lot of oceanic islands are forming through volcanic activity. And if you remember back, we said that Hawaii's kind of neat because it's over a hot spot that periodically heats up, or really starts spewing stuff out, so we can get an archipelago, which is a series of islands, oftentimes islands that have formed over a different period of time. There are other sorts of archipelagos, but Hawaii is a good example of one where the oldest island is maybe 5 million years, and the other ones get younger and younger, and that's great for looking at certain sorts of evolutionary processes. But all of these are oceanic islands. Continental islands, and I'm going to call these continental plate islands. These are larger islands, typically like Madagascar, like Cuba, like New Zealand, that are on their own tectonic plates. Hopefully you're somewhat familiar with the theory of plate tectonics, which I would say is certainly close to as revolutionary for geology as the theory of evolution by natural selection was to biology. And it's just really, it fundamentally changes how you look at different land masses and at islands. And these particular islands are on their own plates and move around and have moved through time. So often they're isolated for long periods of time and not periodically joined up again, at least not like the shelf islands. The continental shelf islands are just high spots on the continental shelf, which is the part of the ocean that's relatively shallow near the edges of the continent. Once you get off the shelf, it dives, gets very, very deep. But because these are on the shelf, when you get low sea level, they can become connected to the mainland again. When you get high sea levels, they get separated again. And this often varies with glaciations. When we get glaciers forming in Antarctica and in lots of mountains and in the Arctic, they suck up lots of water, sea level goes down. These sorts of islands, the shelf islands, join back up with the mainland. Animals can get on those islands. Sea level goes back down when the temperatures warm up. They're isolated, and they can evolve into different species by allopatric speciation, specifically by carians. Lots of neat things go on in these sorts of continental islands, and a fair number of the examples of island evolution that we're going to talk about are taking place on these continental shelf islands. Yet another book, hot off the press. This is from 2010. I just got this in the mail recently. And this just focuses on island mammals. I am a little mammal-centric. There are lots of cool things that go on with birds and with reptiles, because they actually find it a lot easier to get to islands oftentimes, being able to fly or being able to go for longer periods of time without food. But this is a really nice summary of what happens to animals on islands. And it talks about almost all those islands that I showed you before, with the exception of Madagascar. Well, no, actually it does talk about Madagascar too. So it really runs the gamut. Some places that are very diverse, other places that don't have much going on in the way of diversity, but still have some neat processes that are taking place. So check that one out. That's by Wiley Blackwell and just came out this year. A little bit of math. We're going to talk about the MacArthur-Wilson theory of island biogeography. The Wilson in this is E.O. Wilson, who probably needs no further explanation. The MacArthur was really a brilliant ecologist who unfortunately died at a relatively young age due to cancer, I believe. So he was cut off in the prime of his life. It would have really been interesting to see what he would have come up with just as he was hitting maturity in an academic sense. But they came up with this great equilibrium theory that predicts the number of species on any particular island. And there are a few things I want you to keep in mind as we talk about it. One is it only deals with two variables, so it's highly simplified. Reaching equilibrium takes time, and this can vary based on a variety of factors. And species composition can change even after equilibrium has been reached. So this tries to predict the number of species, but that number of species doesn't need to stay constant. So it can change, primarily by colonization or by immigration. And we're going to have a very restricted sense of islands in terms of this definition. In terms of this theory. And that is, an island is a place where the only two processes at work are immigration, receiving species from somewhere else, the mainland, and extinction. Those are the only two ways to add to or get rid of species. The idea is that if we can understand, even theoretically, a simple island like this, then we can look at the other end of the spectrum, a mainland, where species are only generated by evolution, by bifurcation, put those two theories together to get a comprehensive theory that predicts, really, animal diversity, which is a fundamental area of investigation for people like me and for ecologists. Let's talk about colonization first. Here's the graph that we're going to be looking at, and it's a very simple graph. All you need to focus on is the number of species increases to the right. We're going to put colonization on the left, and this is a rate. We'll talk about extinction in a bit, and that's going to be on the right. In theory, we could draw a line like this, such that at 0 species on an island, there are lots of potential species that could come colonize it. So the rate's going to be high. When all the species from the mainland have gotten to the island, you can't add to it. Because remember, our definition is that's the only way they can get there, and so it goes to 0. We could therefore make a nice regression like this and say, OK, that's how it's going to vary with the number of species on an island. And we're not going to put any number at the end there. It doesn't really matter, but just in a general sense. Well, biology is a little more complicated, and the rate is really more like this, at least based on the data that's been collected. And this has to do with the fact that some animals are able to disperse to islands very well, and others have a real tough time at it. Plus, we get different species interactions. So the curve looks a little bit more like this. But the same general trend, starting out high and running to low. Extinction rate is going to be the opposite. When we have no species on an island, on the left, there are no species to go extinct, so the rate has to be 0. When we get many species on the island, then there are many more potential species that can go extinct. And that's where we might see the highest rate of extinction. And so again, we predict a straight line. But like with colonization, it really turns out to be more like this. Because as you get more species on the island, you start getting competition and other factors, which really makes it more difficult to maintain a foothold there. Certainly much more so than if you just go to an island and there's no one else there. These were the two variables then that MacArthur and Wilson thought mostly accounted for the diversity of species, the number of species on an island. So we can graph them both on the same graph, colonization rate, extinction rate, and there's our equilibrium, where the two rates meet. We then follow it down, come up with a number for species, and then we can go to the field and start testing these models. And the way that they tested them, actually I think it was E.O. Wilson and his student Dan Simberloff, I forget, but they went out to really tiny islands down in the Bahamas, where you're mostly getting insects, and they fumigated them. It was back in the 60s when you could do such things. They killed everything on these little islands, and then they track colonization and extinction and look at these numbers, and this is how they tested this phenomenon. I mean, it's really cool, a lot of work, a great reason to have graduate students. Now, we can complicate this a little bit and try to more closely approximate real life, right? Because that's the goal. We want to start with a simple model, see if it works. If it works, start tweaking it till it gets closer and closer to real life. Because what we want to understand is real life. An island size has an effect. Let's say that this extinction rate is for a small island. We would predict that the large island would have an extinction rate more like that. Why? Because a larger island has more space, and it has more diverse habitats. So it can support a relatively larger number of species. So the extinction curve is going to be shifted to the right in a large island relative to a small one. If we compare our equilibrium point for the small one to one for the large one, we're going to see that we end up with more species. And that's what's known as the island size effect. A similar phenomenon occurs with colonization rate. And if you think about this, you could probably come up with what's going to affect that. It's going to be distance. Nearer islands will have a higher probability of receiving animals from the mainland. And even if they don't take the first time they get there, you're going to get lots of other opportunities. A far island, on the other hand, will have fewer opportunities. In fact, some species just won't be able to disperse there because it's so far away. So its colonization curve will be shifted to the left. For the same sized island, then, if we compare our equilibrium point for one that's close to the mainland to one that's far, we'll see that we'd expect fewer species on a distant island than we would on one that's close. And this is something else where we can start looking at real islands doing species areas curves, looking at birds, looking at bats, looking at mammals, looking at insects, all sorts of different things to see how this actually teases out. Because you can only look at one group at a time, right? Because it's really unfair to talk about dispersible ability of birds relative to hedgehogs, for instance. But within a certain group, we've really tweaked and learned a lot about why certain islands have certain numbers of species. A little tough to see, but this is Australia. And Australia is a little bit counterintuitive in this sense, because if we use this definition of an island that we've talked about in terms of equilibrium, right, where species only come from the outside or go extinct, then Australia, even though it's described as an island continent, is not an island. It's a mainland. Because the vast majority of mammals that are there, we'll mostly talk about mammals and other animals, have come from evolution within Australia. This, of course, is accepting the ones that have hitched rides with humans. Because recently, right, we've taken rabbits and foxes and camels and everything else that have really caused issues over there. But prior to that, we think that marsupials, these things that sometimes have pouches, sometimes don't, we think they probably originated in North America, or at least they were there living in North America when the dinosaurs were around. They hiked down through South America, down through Antarctica, and then got over to Australia, and then diversified there into all these different forms that we see today. Things like kangaroos and their relatives, things like possums without an O and their relatives, wombats, a large diversity of animals. So it's mostly intrinsically that these animals have been diversifying. So in this sense, it's not really getting new recruits, and this would be a mainland. Be that as it may, now we're just going to look at some kind of fun examples of things that happen on islands. When we do get animals that get out to islands, what happens? One of the most common phenomena that we see is a change in overall body size. And body size affects everything. It affects reproduction, it affects metabolism, all sorts of things. So it's really fundamental to how an animal makes a living. We see dwarfing, and we see gigantism. A couple examples of dwarfing are elephants and hominids. This is sort of a good segue, I guess, to next week at the museum. Elephants are really good swimmers. There aren't many chances probably that you get to see an elephant swim, but they really are good swimmers. You'll see them out in the ocean. And another way we know this is because they have a really easy time getting to islands that aren't that far away. One example are the Channel Islands, which are off the coast of California. These are two skeletons of mammoths. One is a regular-sized mammoth, and the other is a dwarf. The dwarf one is what we find on the island, and of course, the big one is what we find on the nearby mainland. This also applies, we think, to humans. I'm sure you've all heard of the Hobbit, the Homo floresiensis. This is an artist's reconstruction of the relative size of this animal, this human. I don't know, you can ask Bruce and Scott and Johannes later if this qualifies as a human or not. But we think it's the same phenomenon. Big animals tend to get smaller when they're isolated in smaller areas. And we can talk more later about why this is. No one really knows for sure, but we think big things get small and small things get big. So we can talk about gigantism, which is the other side of the coin, in various insectivore groups, things related to shrews and moles and that sort. This is one of my favorites, Dinogalerics. The dino here is the same root in dinosaur. Dinosaur, of course, means terrible lizard. This means, well, essentially means terrible hedgehog. Although this really isn't a hedgehog, this is a moon rat. It's in the same group. But the skull is on top, and down below is an English hedgehog, right? So not those pygmy ones you have as pets. That's a big one. And those are the skulls. This would have been a 4 or 5 pound hedgehog that is known from the island of Gargano off of Italy. Now, Gargano is part of Italy, but this is one of those continental shelf islands that was isolated for a while, had a very strange fauna evolve, and then joined back up again. Normally, insectivores aren't this big, but when they're isolated, they do tend to enlarge. We do have some that are still alive. These are not Dinogalerics. These are things called Solenodons. Thank you, Joe, for that photo. Solenodons are insectivores that inhabit some of the Caribbean islands. Unfortunately, they're endangered because now humans also inhabit the Caribbean islands. But you get a nice idea of scale for these animals. Really strange things. Small animals get big animals get small. Probably is related to how much food is there, lack of predators, all sorts of other things that are going on. Locomotor changes are something else that we see. Flightless birds is a great example. We have flightless birds on all continents, but you see it pop up a lot on islands. Once a bird gets there, it doesn't want to get blown off again. You see this in beetles, too. A kiwi is an excellent example of a flightless bird of New Zealand. It's there with its egg. If you haven't seen an X-ray of what a kiwi with an egg looks like, you should really look that up next time you're on the internet. It is incredible. And that's a whole story why it has a big egg. But the important point is it's flightless. As is another less well-known bird, the kakapo. Kakapo is just really interesting. It's a parrot. It's one of at least, I think, 3 genera that are present on New Zealand. Remember, New Zealand is one of these continental plate islands, so it's been isolated for a long time. It's the largest parrot, probably one of the longest-lived parrots. It's a nocturnal parrot, which is strange, and it's a fruit eater. And these things weigh something like 9 to 10 pounds. Just to put this in perspective, I was at the museum recently. They're doing one of those live animal shows, and they had, actually, I think Peter was doing a live animal show, and you had a vulture there, and I think the vulture weighed about 3 pounds. Is that about right?
About 5.
5 pounds. So twice as big as a vulture. This is one big parrot. There are other cool parrots there called kias. They cause lots more problems because they're omnivorous and they have big beaks and they're really curious. But the kakapo is flightless. This is something you can do when you're on an island, when there aren't any predators to worry about. We can get this also occurring in mammals, terrestriality. And this isn't obligate terrestriality. This is facultative terrestriality in a really interesting group known as mystacenans. It's a special group of bats. Bats are the second most successful group of mammals in terms of the number of species. And there are 2 species on New Zealand, and they look something like this. This is a male. And they forage a chunk of their time on the ground, which again is not something you see in lots of other places with lots of predators. And I did happen to pull up just a little bit of a movie here. Hopefully you can see a little bit what these things look like. I really love bats, but it's a little creepy. The only other bats that we see do anything like this are the vampire bats. And they're not foraging around like this. They're just landing to get close to their prey items, essentially. And if you like David Attenborough, there's an excellent series of videos called The Life of Mammals. And it's on the first DVD, I think it's the 3rd one, Insect Eaters. And at the end, he has a great series on swarms of these mystacenans in New Zealand. And it's really something impressive to see. So go buy that one or check it out at your library. It's really pretty cool. We can look at things on a community level. As I said, lizards often get to islands, and we have little ones called anoles that are very common here in the Western Hemisphere. They're often called chameleons, but they're not. They're a separate group. There's a picture of one up there. And people have done a lot of very interesting studies, essentially looking at the sorts of habitats these are adapted for, whether they hang out in the trees, whether they hang out on the ground, whether they're large size. And up top there, we have a phylogeny, a family tree. Don't worry about the details. What I want you to notice is that these patterns, which represent different ecomorphs, so different morphologies correlated with different habitats, are scattered, such that we don't have one clade, one group that's all living in the trees, one group that's living in the ground. Rather, these different ecomorphs can evolve from all sorts of different ancestors. Right? They're scattered throughout the tree. And if we look at different islands, Cuba, Hispaniola, Jamaica, Puerto Rico, we see that each island has the 4 main different ecomorphs, which we'd expect if they have the same niches, but they're all evolving at different parts of the tree. So it's not an instance of the same animal going to different islands. It's really one animal getting there and then filling these different niches. So we get broadly similar patterns on these separate islands, and it's like running 4 different experiments. You can't do this. This happens over evolutionary time. And islands enable you to do virtual experiments that you can't do in any other way. I think these things speak for themselves. Hoplotomyrix, a 5-horned cervoid, so it's a relative of deer and elk and things like that. these are not antlers. These are actually horns. They probably would have had a covering. These weren't shed. This also comes from Gargano. Gargano being the home of Dinogalerix, the terrible hedgehog and the terrible moon rat. Candia cervus, which is a true deer. It's small. We see island dwarfism, but this huge, strange antler that just comes off like looks like someone stuck a broomstick on there. Just bizarre, almost as long as the body. This is from Crete. These islands in the Mediterranean often were isolated and then joined up again, so we see lots of different animals like this evolving through time. Really lots of neat things going on in Crete and Gargano and in that area of the world. But I'd like to finish up by just highlighting the importance, in a broader sense, of these island studies. Because we think of islands as surrounded by water, but really lakes are also islands, because they're isolated from other areas from which they're going to get species. And mountaintops are also islands, and the habitats on mountains, animals dispersing from one to the other. And fragmented habitats are also becoming islands, patches of forest in the Amazon. So essentially, islands are everywhere. And it's only by understanding these isolated populations, these island processes, that we can really get a handle on the factors that affect species diversity. And with that, we have to take a look at the fossil record. We have to look back at information like this, all these strange animals in the past, because it's only through looking at evolutionary time that we can understand these isolated island processes and really get a handle on how did we end up with this really magnificent, what I think is fascinating, diversity of mammals that we have today. And with that, I'm looking forward to dinner. We'll be happy to answer any questions.